Yeah, we can.
Good, thank you. We left up on this slide here talking about membrane protein structural classes and functional classes, and we'll talk about this all in more detail as we move along. So membrane carbohydrates, glycoproteins plus glycolipids is our glycocalyx. This is on the E face of the cell. And you can see that here. Anytime someone shows you a membrane, on the top would be the extracellular, or you could say interstitial space, but that would be the E face, and then on the bottom will be the P face. If someone flips it upside down, that's not cool. It should be like this. So we don't have these sugars on the inside of the plasma membrane, on the outside. Collectively, we call that the glycocalyx. It's a lot more elaborate than what's shown here. It's actually more elaborate than what's shown here too, but this is carbohydrates here, or these are. And these carbohydrates here are there for many reasons. So the glycocalyx can be there to prevent, mechanical injury to the cell, chemical injury to the cell, but also these carbohydrates serve as identity markers. So these are the identity markers that identify a cell as either bringing self to your body or foreign to another body. And you can see that here. This is a cell that has these antigens on the surface of it. So these carbohydrates attached to proteins or lipids, and then antibodies will bind to these, and therefore these sugars are antigenic. And we'll talk about that when we talk about blood typing. So that's the anatomy of the cell. So let's talk about the physiology. So the general principles, the plasma membrane is inherently selectively permeable. What I mean by that is if you just have a lipid bilayer, no proteins in it, nothing, then it's still selectively permeable. It's only going to allow things to go through that are small, uncharged. Things that are large, things that are charged can't get through the membrane. Gases can get through the membrane. And again, no proteins at all. So it already has this selective permeable nature because those polar head groups on the outside are attracted to water, but the fatty acids on the inside create this hydrophobic core. If we want something to move across the membrane, then it requires two things. And this is really important for like the rest of the semester. So we need a pathway and we need an energy source. And you might say, well, you just talked about the membrane. Well, the pathway might not be the bilayer. The pathway might be a protein in the membrane or something like that. Regardless of what the pathway is, we're going to need an energy source as well. And so this can be passive or it can be active transport. If it's passive, then you're going to be utilizing the energy of a concentration gradient. So you need concentration gradients so that things will move either from the inside of the cell to the outside or outside to the inside. For active transport, you're going to be using the energy of ATP. So now we use the energy of ATP, hydrolysis, to power something to move against this concentration gradient across the plasma membrane. Now, there's things that will affect the rate at which something moves across the membrane. One would be distance, and that's really important. So the way I would like you to write that down is diffusion is distance limited. So if you have a bunch of X's right here, and let's say they're glucose, and they're going to diffuse in this direction, well, eventually they're going to be 2, then 1. So they're just going to dissipate because some are going to cruise off this way, this way. So the gradient gets smaller as something diffuses. And so diffusion is very distance limited. And that's why all cells of the human body are within 2 cell lengths from a blood vessel. And we know that a cell length is about 10 microns. So capillaries are within 20 microns from any cell of the human body because diffusion is distance limited. We can't diffuse over long distances. Now, there are examples where cells are dead because diffusion is too far away, and that would be like the surface of your skin. So the surface of your skin, those cells are not alive, and it's largely because they're way far away from the vasculature that would supply blood to them. The size of the molecule will dictate how fast it moves. Larger is slower, how steep the gradient is. So if you got more X's here or less X's, the concentration gradient would be stronger or weaker, respectively. We know that with temperature, that increases the rate of anything. And then electrical forces is a really important thing. So if we have a membrane and we have a bunch of X's here, and we want X to move in here. If X happens to be positively charged, and the inside of the cell is negatively charged, then two things will be drawing that X in. One, the concentration gradient. Two, the charge. So there's an electrical force that's favoring its entrance into the cell as well. And we'll get into that in more detail when we talk about action potentials. So this is Fick's law of diffusion. Quite honestly, I never ask questions about it, so don't worry about it. It's just saying that the diffusion rate of something is directly proportional to the permeability of whatever solute you're talking about, the area of membrane, the concentration gradient, and the only thing it's inversely proportional to is the molecular weight of the solute. In other words, larger things diffuse more slowly. So this is a situation where you don't have a gradient of solute, and now you have a very steep gradient. And we say that these molecules or solutes can move downhill, and that would be just passively. And if they were to move uphill, that would be actively. So you need ATP to drive them uphill. Now, keep in mind that down and up have nothing to do with the orientation of this image. If I turn this image to its side, then still moving from the left to the right would be down the concentration gradient. In other words, downhill just means from larger concentration to smaller concentration. So if I were to draw these X's here, membrane, 1X here, that's still downhill that way. So what ultimately drives this? Well, basically the second law of thermodynamics, everything tends towards disorder and Brownian motion. So we have these molecules that are just bouncing off one another and moving as far away from one another as they possibly can. Okay, so let's talk about passive transport first. Let's look at simple diffusion as a mechanism of passive transport. And this just means things are going right through the plasma membrane. Now, there's your pathway, but now what's the energy source? The energy source is a concentration gradient. So you have a concentration gradient for these small nonpolar molecules to move across the bilayer. So here's a good example right here. And you can see that fatty acids, steroids, gases like O2, CO2, nitric oxide, we'll talk about all of them this semester. They just blow right through the plasma membrane. And you may think a steroid's a pretty large molecule. It is, but it's flat, so it can get through the bilayer. And fatty acids can get through, but triglycerides can't. They're too big. Monoglycerides actually can get through as well, but diglycerides can't. And we'll talk about that later as well. Osmosis is a special case of diffusion in which what's diffusing is water. So water is moving down its concentration gradient. It's moving from a higher to a lower osmotic potential. So that's interesting. Normally, when we think of something diffusing, we think about it diffusing in water. Now we're talking about water molecules themselves diffusing. So this is one thing you want to remember, that a solution with many dissolved solutes, water is always going to want to go in that direction. So water likes to follow solute. So in other words, if you put a lot of glucose in your blood, more than your kidneys can reabsorb, then glucose is going to end up leaving your body through your urine, and it's going to take water with it. You're going to become dehydrated. And that's because water likes to follow solute. Wherever there's more solute, we call that a low osmotic potential. I know it's kind of counterintuitive, but low osmotic potential means there's a low potential for water to leave that environment by osmosis. And the reason for that is there's a lot of solute there. So wherever there's more solute, there's a lower osmotic potential. So take a look at this experiment here. We have this flask here, U-shaped flask. On the A side and B side, there's glucose and there's water. Glucose is in purple, water is in blue. They're actually showing you individual water molecules in blue, which is really cool about this. And then they got a semi-permeable membrane here. So I'll give you an experiment like this on the exam. And the semi-permeable membrane, I have to tell you the nature of it. In other words, what is it permeable to? What is it impermeable to? This is permeable to water, but not permeable to glucose. And so someone sets up this experiment with a certain solution of glucose on one side and on the other side. So who's got the lower osmotic potential, A or B?
B.
You got it. So B's got the lower osmotic potential because it has more glucose molecules, right? And so water wants to go that way. And quite frankly, it will. If you look at, say, this is time equals 0, this is time equals 1, water is going that way. As a matter of fact, the amount of water is increasing on B side because water is following glucose. Now, back at time equals 0, glucose wanted to go from B to A, but it can't because the membrane's not permeable to it. Another way of looking at this, people, is literally what's happening here is you could say, well, water is following the glucose, right? Or you could say water is going where there's a lower osmotic potential. Another way of looking at it is look at the water molecules here. There's many more little blue dots there than here. Water is diffusing down its concentration gradient, right? So wherever there's more solute, I don't care if it's glucose or whatever it is, wherever there's more solute, there'll always be less water molecules and vice versa. So water is just diffusing down its concentration gradient.
Can I ask a question real quick?
Yeah,
Okay, so you said more solute equals a low osmotic potential, correct?
correct.
So you were saying we said B has more osmotic potential, but doesn't it have more solute?
No,
Like,
no, B has a lower osmotic potential.
oh, okay, but okay, lower. Okay, got it. Okay, I heard that wrong.
Yep,
Thank you.
no problem. Now, when I was sitting where you were, like, not where you were like right now, but like where you were like in terms where you are in terms of at Wayne State, like literally, maybe even a year before you, 2 years before you, when I was sitting in 1510 or cell bio, my prof was telling me that in General Lectures 150, by the way, my prof was telling me that the main way water got through cells was the plasma membrane, just like this. They said, hey, there's water and it gets right through the membrane. And we said, well, how does it do that, right? It's polar, and these are all polar head groups here and here. So how's water getting through the membrane? And they just said, well, once it breaks past this little polar area here, or once it engages here because of the polarity, it's able to zip through. And we just said, okay. And we just accepted that. The reality is water does get to the through the membrane to some extent this way because the membrane's fluid. And so some water can seep through the membrane, but the vast majority of water does not go through the membrane this way. And there was a Nobel Prize given for the individual that actually discovered much of the water, most of the water goes through another mechanism. We'll talk about that soon. Let's do a question on this. Um, C6. Okay, good. So let's talk about tonicity. So the cells of the human body are bathed in a solution that's called interstitial fluid. You know that. Well, that fluid has basically the same concentration of solutes on the outside as in the inside of the cell. So there really is no net new movement of water. There is some movement of water into the cells and out of the cells because when ions move in and out of cells, that changes the concentration gradients a little bit, but there's no net movement. And so we say that our cells are in an isotonic environment. That means that ICF is isotonic to ISF. And these are red blood cells that are shown in an isotonic solution. So that's what a red blood cell looks like. That's scanning electron microscopy there. So here we have a cell in a hypotonic solution. So in a hypotonic solution, hypo means low. That means the solution has low solute relative to the cell. So who has the lower osmotic potential, the cell or its solution?
The cell.
Right. And where does water want to go? You could
Right.
say it two ways.
Water wants to go in.
Osmotic potential.
It wants to go where there's more solute, or it wants to go where there's a lower osmotic potential, right? So water is going to go into the cell. And what's going to happen when you place a cell in a hypotonic environment, it's going to swell. And potentially what it's going to do is lyse. So the cell will just burst open as a result of that. The flip side is if you put a cell in a hypertonic solution, think of hyper as a lot of things moving around, so a lot of solute moving around. So the solution has much more solute than the cell. Who's got the lower osmotic potential?
The solution.
The
The
solution.
solution. So water wants to go there, and water is going to go there, and then the cell is going to shrivel. It's going to shrink. There's a red blood cell shriveling up. We call that crenating. The cell has crenated. Now, normally our cells don't do this. Normally our cells don't swell, and normally they don't shrink. Under abnormal circumstances, they can. I'll give you an anecdote about this later. We'll talk a little bit about water intoxication, where if you drink too much water, basically the neurons in your brain can swell. And when they do, they don't fire right. Now, what you could say to someone is, what happens to a cell? And by the way, one way I like to do this is I like to write hypotonic like this. You could do that if you want. And then it swells, right? What happens if a cell is hypertonic to its solution?
Doesn't that mean the cell has more solute in
Right.
it?
So
Okay.
what happens to it?
So it's gonna
It would
swell.
shrink,
If the cell's hypertonic to its solution,
so it would shrink.
nope. As Angelina said, it would be just like this. If the cell's hypertonic to its solution, it's got more solutes than its solution.
I might
So
have just
saying,
misheard. Okay, yeah.
yeah, so saying a cell's hypertonic to its solution is the same thing as saying a cell's in a hypotonic solution.
Oh, okay. I was thinking, I was like flipping it, like saying like,
Yeah,
okay, nope,
and I
never
was
mind.
flipping it just to try to trick you guys.
Yeah, I was
More
gonna
often than
And it
not,
worked.
yeah, more often than not, when someone talks about it, they talk about the cell in reference to its solution, or sorry, the solution in reference to the cell. So the
You said
cell's in
this.
a blank solution. It's in a blank solution.
You said the cell is hypertonic to its environment.
That was the example I gave. What if a cell
Wou-
is hypertonic to
Wouldn't
its
that mean that it's in a hypotonic solution?
I'm sorry?
That would mean that it's in like if it is hypertonic to its solution, then it would be in a hypotonic solution, right?
Correct.
Okay, I got it now. Thank you. I
Thank
I
you.
hate these questions.
Indeed. Yeah, well, expected on the exam.
I every time I've ever had one of these on an exam, I have to sit there for like 5 minutes.
All right, let's look at facilitated diffusion, movement of ions and large polar molecules down their concentration gradient across a membrane facilitated by membrane proteins. So this can be facilitated by channels in the membrane that we often call pores or gated channels, carrier proteins. And we were talking about those when we were talking about the different classes, the functional classes of proteins. So here's a really good example of some facilitated diffusion. And so these are carrier proteins here. And the carrier proteins can be uniporters. That means they carry one solute in one direction across the plasma membrane. They could be co-transporters, which means they carry two or more solutes. Doesn't have to be just two, but two or more in the same direction across the membrane. That subclass would be symporter. Or they carry two or more solutes in opposite directions across the membrane. And that subclass of co-transporter would be an antiporter. And we'll talk about plenty of uniporters, symporters, and antiporters in this class. These facilitated transporters exhibit specificity and saturation limitations just like enzymes. For example, you have transporters that are located in your kidney for you to reabsorb glucose. So when we filter our blood, we filter glucose that would potentially end up in our urine, but the amount of glucose that ends up in our urine is zero. So it's on its way to the toilet, but it never ends up there because we have these facilitated transporters that reabsorb the glucose. Well, the problem is there's only so many of these transporters there. So if you're a diabetic and you're not taking your insulin, then the glucose load in the kidney, meaning the amount of glucose, is really high. And so it'll go through all these transporters it can, but once those transporters are moving as the maximum amount they can move, the rest of the glucose just spills in the urine. And that's diabetes. That's by definition diabetes. Now, probably if you ate a couple large pizzas, you would spill a little glucose into the urine as well, and that wouldn't be abnormal. Now, this is the Nobel Prize here, 2003, really not that long ago considering science, because you still barely find these in textbooks nowadays. But aquaporins, that's what turned science on its head. That's what profs didn't know when I was sitting in your seat. So they didn't know that there were water pores that water travels through the membrane. So look at a little bit of water going through the bilayer. The vast majority goes to proteins that are called aquaporins, and there's a whole family of aquaporins that have now been discovered. So Peter Agre, 2003, he actually shared this Nobel Prize with somebody else that I'll tell you about in a little bit. And that one won't be on the test. This one's fair game, the other one won't. So we have leak and gated ion channels. So channels could be always open. And if they're open, they have high specificity. So there's going to be a certain ion or ions that move through it. Normally it's just an ion. So there's going to be sodium going through it or potassium, chloride, whatever it may be. So we'll call them sodium leak channels, potassium leak channels. Potassium leak channels are a big deal in this class. And you might say to yourself, man, that just seems so like random to have things just passively moving with no regulation. You'll see that it is indirectly regulated, but without potassium leak channels, we would not exist. So I'll explain that in more detail later. Big ones here, we'll talk about this a lot. You've probably learned about neurotransmitters in other classes. Well, this is what they bind to. They bind to ligand-gated channels. So these are basically receptors that are closed normally. And then when ligand binds to the receptor, which is a neurotransmitter, and this one requires two neurotransmitters to bind it, same ones, probably acetylcholine. And when acetylcholine binds, what happens, it causes a conformational change in that protein such that it becomes a channel, and moreover, a channel permissive to a specific ion. It's just amazing. So acetylcholine binds, it becomes a channel, and it becomes a channel permissive to a particular ion. This one's sodium. So we would call this a ligand-gated sodium channel. We have ligand-gated chloride channels. We have ligand-gated calcium channels, so on and so forth. Big one in this class will be a ligand-gated sodium channel, indeed regulated by acetylcholine.
Is that for muscles?
In muscle and many other places, but absolutely muscle, my friend. We also have the voltage gated channel, which is going to be very important in this class as well. And I think the coolest of all the three. So these are the three ways we gate channels. And so here, the protein is normally closed. It's got this little ball and chain mechanism here that is closing it. And what happens is you don't need no ligand binding to it or anything like that. You have a change in membrane potential. So as I said, I believe before, cells are normally more positive on the outside than the inside. But if you change the membrane potential and you make the cell more positive on the inside than the outside, then this protein now, and let me just change it right here instead of over here. So now you make it more positive, sorry, on the inside. Remember that this ball and chain was feeling negative charge on the inside. It's now feeling positive charge. And that ball and chain is nothing more than a string of amino acids with R groups. So those functional groups were feeling negative charge, which caused that ball to lodge into that protein on the P phase. But now when there's positive charge there, you might say, well, how did the cell become more positive on the inside than the outside? Well, way down here in the membrane, someone opened a channel that let sodium come in. And that sodium building up caused that positive charge. And as a result of that, now this thing moves out of the way, and it happened to be a channel permissive to potassium. And now potassium leaves the cell down its concentration gradient. And you might say, well, why would potassium leave? Well, it's because there's more on the inside of the cell than the outside of the cell. And you might say, why? And we will answer that soon. Sorry, I said that these were the three ways that we gate. We don't, obviously lead channels are not gated. The three ways we gate are ligand gated, voltage gated. This is the last one here, mechanically gated channels. And so this is a mechanical deformation of the membrane that actually pries open the channel and makes it permissive to ions. Tell me an example of that we've talked about.
Like pinching your hand.
Was it in a,
I know one from my other class, hearing, like how
No,
we hear.
I want it from
Oh.
hearing you right though,
was
but
it
I
in
want,
Child
parturition?
labor.
yeah, give me the specifics of child labor.
In parturition on the cervix, there were the like the receptors that were like sensitive to mechanical stress.
Yep. So the mechanically gated channels, the fetus head pushing against the cervix, pries open these channels, ions come through, you get a depolarization and you get action potentials that propagate along afferent neurons back to the brain. All right. So this is the other part of the Nobel Prize in 2003 that you don't have to know. It's just really insanely cool that this person discovered this. Basically, what this guy discovered, Roderick McKinnon, and literally these two people shared the Nobel Prize in 2003, was the specificity of the potassium channel. It's like, hey, why is the channel specific to potassium? Why can't sodium go through it? And as a matter of fact, you might believe, if you take a look at sodium, that it's also positively charged and is smaller in angstroms than potassium. Why in the hell can't it go through it, right? It's actually smaller and it's got the same charge. Why can't a sodium ion go through a potassium channel? And so what he found was something that he called a selectivity pore. So the channel has a selectivity pore. And there's a bunch of carbonyl atoms that are facing the inside of this pore in the membrane. This would be, in the bilayer of the cell. And you can see there that potassium is large enough that it can form electrostatic interactions with these oxygen atoms, these ketones here, but sodium can't. And since sodium can't form electrostatic interactions with all four of these, it can't move through the channel. So what he discovered was that the mechanism by which the voltage gated potassium channel is selective to potassium and why it can't allow sodium. And what he discovered is what? Sodium's to what?
No.
Yeah, right. Everyone was like, why can't it go through? It's smaller than potassium. And he said, because it's too small. And again, if you don't get this, no big deal. I think this is really cool. It's just for your personal edification. Glute transporters are a big deal. We'll talk about them all semester. We call them glutes. So here's one right here. This is facilitated diffusion. Glucose is a polar molecule. It's got a bunch of hydroxyl groups, can't go through the membrane, and it's too big as well. So what happens is it binds to this protein in the membrane called the glucose transporter. When it binds, the protein undergoes a conformational change. And the conformational change is that it opens on the inside of the cell. And the reality is when it opens like this, it causes another conformational change that causes the binding affinity for glucose to that pocket to reduce. So there's a couple conformational changes going on there, and then glucose gets dropped off. But again, you still need a concentration gradient. So this will facilitate, but you need an energy source. So you need more glucose on the outside of the cell than the inside. So we talked about some glutes before, and you don't have to know all of these. I do want you to know GLUT 4. We said that that's an insulin sensitive transporter that's in fat cells and skeletal muscle cells. We said that GLUT 2 we found in the liver. It's getting glucose all the time. It doesn't need glucose transporters to be put in the membrane via insulin. And then the other one is GLUT 1. GLUT 1 is pretty much in every cell. Every cell can get a little bit of glucose. So it's like the basal transporter in most cells. But don't worry about, GLUT2 and 5 in the intestines and GLUT2 and pancreas. Don't worry about that. So there's glucose coming into the cell through a glucose transporter. This is a myocyte, a muscle cell. And look what happens. The first thing that happens to glucose, it gets phosphorylated to glucose 6-phosphate. Why?
Is that so that it can be formed into glycogen?
Yeah, so that's one of the reasons. So one of the reasons is that's the priming step for
Isn't
a
it to
bunch of
trap it?
glucose. I'm sorry?
I thought it's to trap it, right?
Well, I'm
Am I-
getting there. So you're
Okay.
correct. So one of the reasons is to prime it to form glycogen. So you're going to get a bunch of glucose monomers that are formed into this polymer. This is glycogen. It's a branched polymer of glucose monomers. And as the other individual was saying, the other reason is that not really, so to speak, to trap glucose, but kind of correct. But here's the deal. Once you make a glucose 6-phosphate, it's not glucose anymore. And that's important so that you still have your concentration gradient so glucose can come in. So if you just got glucose coming in and glucose just hanging out as glucose, you're going to lose your concentration gradient. But the
Is
other
it?
problem is if you got glucose floating around in the cell, what's that going to do?
It'll undergo glycolysis.
Not that. I mean, it can, but instead of glucose being combined into glycogen, if you have a bunch of glucose monomers floating around, what would that cause?
It can get to the lipid bilayer of the cell membrane again.
It would do what?
It would pass to the lipid bilayer again. Like,
No, it won't pass through.
Diabetes?
Would it
Not
create a
diabetes.
hypotonic environment?
Yeah, so it would create a hypertonic environment. Then what would happen?
The cells would lyse.
Yeah, water would come in and the cell would lyse. But by making it into glycogen, that doesn't contribute to its osmotic potential. So yeah, a couple reasons why we convert glucose into glycogen. Now, there's a way to upregulate glucose transporters in the membrane in the absence of insulin, and you can do that, people, by exercising. So there's a mechanical phenomenon, and I've done honors projects for 2 semesters at Michigan State, and no one can find out what the deal is behind it. It's believed that muscle contraction, the mechanical force of muscle contraction, can cause the upregulation of glucose transporters, and that's why exercise is like the #1 prescription for diabetes or obesity or whatever it may be, because you can improve your glucose metabolism and your blood sugar by exercising merely because you can increase the amount of GLUT transporters put in the membrane. Literally causes an upregulation in GLUTs.
I have a question.
Yeah,
So, I just want to make sure I got it right. The reason that glucose gets converted to glycogen, or like some of the reasons, is to maintain the concentration gradient, and so that the cell doesn't like water doesn't start coming into the cell.
correct. Both of
Okay.
those are true. And then you could also say, well, really the cell wants to store it as glycogen so that we can use the glycogen later. But yeah, really it's stored as glycogen instead of stored as glucose, so it doesn't contribute to the osmotic potential.
Thank you.
Indeed. All right, active transport. So we're done with passive, we're talking about active. So we're gonna look at primary active transport and secondary active transport. So here we got these movements of solutes or ions against their concentration gradient. So again, the pathway is going to be some kind of protein in the membrane, and then the energy source is actually going to be ATP. And the best example of that is the most important protein on the surface of this planet, that is the sodium potassium pump. And this is one of those examples of something that'll be on like every exam. So the course is not cumulative in the strict sense of the word, but this is so important that it's going to be on every exam, the sodium potassium pump. So what is it? Well, it's a protein. It's an antiporter because it's actually transporting 3 sodium outside the cell for 2 potassium in. So 3 sodium out, 2 potassium in. You could say it's a co-transporter. You could say it's an antiporter if you like. And it does that exchange of 3 sodium for 2 potassium by hydrolyzing ATP to ADP and P. That's why we call it a sodium potassium ATPase, because it's a protein that can not only translocate these ions, but it uses and hydrolyzes ATP and ADP and P. Now, it's moving sodium and potassium against their concentration gradients. And then you might say, why? Because the sodium potassium pump is what creates these concentration gradients. So the sodium potassium pump is pushing sodium on the outside of the cell and potassium on the inside, but it keeps running and it keeps pumping more sodium against sodium and more potassium against potassium on the inside of the cell. So it has to use ATP to do this. And these things are running all day long. So that just an example here of kind of remembering that sodium on the outside, potassium on the inside. Here's the importance. 1/3 of all the reason why you eat is just to run sodium potassium pumps. 1/3 of all body energy is used just to run sodium potassium pumps. So they're running all day long. And their goal is to put more sodium on the outside of the cell than the inside and more potassium on the inside than the outside. So suffice to say that no matter what, anytime you're thinking about a cell, the cell has more sodium on the outside than the inside and more potassium on the inside than the outside, always. Now, the reason why that's so fundamentally important is everything that you can possibly believe that you can do. So, walk, think, talk, smell, see, everything that you can possibly imagine that the human body can do is the result of sodium coming in cells and potassium leaving. Everything boils down to sodium and potassium out. So that's how fundamentally important this protein is.
Professor, you said potassium leaving?
Yes. Sodium out, potassium in. But what I'm saying is, and thank you for bringing up your inquiry. Once sodium's on the outside of the cell and potassium's on the inside of the cell in these huge gradients, then we're going to let them come in and out of the cell respectively against their concentration gradients. So now that sodium's on the outside of the cell, for example, acetylcholine will bind to a ligand-gated sodium channel. Well, now that the channel's open, you have a pathway, but why will sodium come in the cell if you do that? Because there's a gradient. If you open up a ligand-gated potassium channel, why will potassium leave? Because there's a gradient. And so that's what I meant by them coming in or leaving. Let's look at secondary active transport. So we have these molecules, ions, whatever they may be, moving against their concentration gradient again. But they're actually driven by the energy of a concentration gradient. And again, facilitated by a carrier protein. So probably the best example of secondary active transport would be an SGLT, which is a sodium glucose transporter. So imagine this being your small intestine. We got cells that line the small intestine that have microvilli. And that's what we're looking at right here. So we're just kind of blowing this up and looking at that. And so the inside of the small intestine right here, we call the lumen. So that's the inside of the small intestine. There's that term lumen for you. And what happens is you eat glucose. It gets into your stomach. It gets into your small intestine. And the glucose is going to come into our intestinal lining cells. Anyone kind of look at those cells I asked you to, not memorize, but kind of have an idea of? What do we call these cells that line our small intestine?
Simple squamous cells.
They're actually simple columnar cells. Enterocytes or enterocytes. So these enterocytes, and we'll talk about that more later, in their apical border, apical means the border of the cell that is facing the lumen. Basal side of the cell is what faces the blood or the connective tissue. In their apical border, they have these microvilli. And in the membrane of the microvilli, they have enzymes. And one enzyme is maltase that can cut, for example, maltose up to into glucose and glucose. Well, then how does glucose get into the cell? Right here. That's the SGLT. So what is it doing? It's using the concentration gradient of sodium to drag glucose in because there's more glucose on the inside of your cell than the inside of your small intestine. So you have to bring glucose in against your concentration gradient. So this is kind of like what's happening here. Actually, I'll come back to that in a second. Why is there more sodium on the outside of the cell than the inside? Right here, sodium potassium pump. So it's pumping potassium in the cell and sodium out. So you're always going to have this gradient here, and this is the way that it works. So what happens is you have the sodium potassium pump pumping sodium out, pumping potassium in, and then the sodium that was pumped out, that's a potential energy. So it's like energy of position, and you can use that energy because sodium wants to come back in. And if sodium comes back in, that kinetic energy, that energy of motion now can be used to drag something against. Let's say that's a bunch of glucose here against its concentration gradient.
I have a question.
Yeah.
So at the basal surface, wouldn't you use a GLUT transporter to take out the glucose from the intestines?
Yeah. So we actually probably won't even get into that in this class, but yeah, there'd be a glute here to get it into your blood.
Oh, okay.
I think it's a glute 5 or something like that, but I don't remember. So where do we find SGLT1s? We find them in the small intestine. This is an SGLT1. Where do we find SGLT2s? We find them in the kidney. So you might say to yourself, what do you mean SGLT2 in the kidney? I just said earlier that if you eat 2 large pizzas, you probably would spill some glucose into your urine. And the reason for that is you would overcome these. You only have so many SGLT2s to allow you to reabsorb glucose by secondary active transport. So why do we call it secondary and not primary? Because this right here does not directly use the energy of ATP. This does. This, so to speak, secondarily uses the energy of ATP. This is a primary active transporter. It uses the energy of ATP to do its job. And now this byproduct of using ATP, sodium gradient, is used to drag glucose in.
I have a question.
Yeah,
So just to clarify, secondary active transport is like reliant on the concentration gradient for
100%.
primary active transport, so it wouldn't be able to occur without
Yep.
primary.
Nice
Okay.
nice way of putting it. It is
Thank you.
100% reliant on primary active transport.
Thanks.
Mhm. Yeah.
So this might be a silly question, but so I understood everything up to like why is glucose dependent on sodium? Like if the concentration gradient inside the cell is higher, why like if it's passive also, like why are they dependent on each other?
Well, if you got more glucose in the cell than the outside, then glucose can't come in by itself because that would be moving against its concentration gradient. So it needs an energy source to come in if you want to move it against its concentration gradient.
So sodium is the energy.
You got it.
Okay, all right, thank you.
Indeed. All right. Let's do a question about that. I think I got a interesting one about this. Yeah. Yeah, I like both of these questions. Feel free to ask any questions during this time too. A lot of people are confused on secondary active transport, so you can't ask enough questions about it. This is a prototype example of an exam question here.
Professor, I have a question in regards to hypotonic and hypertonic states.
All right, give me
Um,
1 second on that. Let's get this concept out of the way first.
Mhm.
Actually, that one might just save that one for office hours after.
All right.
Very good here, people. One more. Another great example of an exam question. Nice catch, by the way, on the last question about integral membrane protein. So that's a tough question because, everything that I'm asking is not just on one slide, right? So I'm talking about how much energy is used. You have to know the 1/3 thing. And then I go way back to 10 slides earlier where we talked about the difference between peripheral and integral membrane protein.
I have a question. So are symport and symporter and antiporter still used when you're talking about active transport? Because my understanding was that that was only used when you were referring to uh or like ATP powered, I guess. Because my understanding was that those were the ones that used like the concentration gradient of the other of its a co-transported molecule to drive it. Like those are the only ones that were symport and antiport, and like the sodium potassium pump like wouldn't be an antiporter as a result.
Yeah, both active and passive are symporters and antiporters.
Okay. I'll I'll correct myself on that then.
Professor, just a clarification, the glucose is still absorbed in the intestine, right? By secondary active transports.
You mean still versus what? Did we
Like,
say that it wasn't?
no, I mean, I'm just reading this question here, the first one, the mechanism of which glucose is absorbed in most body. It says except the intestine, but it is in the.
Oh, yeah. But what does that one mean? Anyone know? So it's a very good, I'm glad you brought that up. So here's the deal. Glucose is absorbed in the small intestine through SGLTs. How about the rest of the cells in the body?
By GLUT1.
Close enough. Yeah, well, it could be GLUT1s. You're correct. GLUT1s, GLUT4s, whatever, glutes in general. Yeah, and I'm glad you actually chose GLUT1 because that's the basal one, right?
Yep.
Good.
Um,
Any other questions on this one here?
I have a
Yeah.
question.
Yeah.
Is the reason B is incorrect because it says directly uses the
Yes.
energy of ATP? Okay.
Yep. Indirect. Another one?
No, I thought for a second it was like the first one I was asking for the incorrect, but then I realized other ones were incorrect too, so I picked B, so I was gonna ask about that, but thank you.
Mhm. Okay. So this is the pump leak model. So what it says is this, and really it just ties this all together. What it says is if you pump something outside the cell, then you can allow it to leak back in the cell and do things. So here's our sodium potassium pump. We pump sodium out, and now we have this huge gradient of sodium outside the cell. What can we do with it? Well, we can let it just leak right back in. Now, not necessarily just leak, it could go through sodium leak channels, but it also could go through ligand-gated sodium channels, voltage-gated channels, mechanically gated channels. So for example, parturition doesn't exist if a sodium potassium pump doesn't exist. Because when the fetus's head was pushing against the cervix and it opened those mechanically gated channels, the pathway was created, but there would be no energy source or there'd be no gradient, right? You have to have a gradient for sodium to come in, and sodium will come in so long as there's more on the outside of the cell. And the deal is, there always is. There always is more sodium on the outside of the cell. And that's why sodium potassium pumps are cranking all day long. What else? Well, we can use sodium to drag something else in the cell, like amino acids, like glucose, whatever it may be. So sodium glucose transporter, sodium amino acid transporter, so on and so forth. We could drag something out of the cell, sodium hydrogen exchanger. We could drag calcium out of the cell, sodium calcium exchanger. So that's really what the pump leak model is. I wouldn't worry about the rest of this stuff here. That's what I really want you to know about this slide. Yeah, we have calcium pumps and proton pumps in the body as well. And we'll talk about those, but that's the gist of the pump leak model. Pump out something and you can use its gradient to do things. Vesicular transport, I'm not really going to go through this. You guys know about pinocytosis, receptor-mediated endocytosis, phagocytosis. Please know that LDL, that's the way that it gets into cells. So there's actually receptors for LDL. So this is low-density lipoprotein, which, by the way, is not cholesterol. It's a carrier for cholesterol. And then when the receptor binds to it, it goes through receptor-mediated endocytosis. And the reason I want you to know this is for when we talk about atherosclerosis later, when we get to cardiovascular physiology. This is phagocytosis and antigen presentation. So here's a phagocytic cell that's phagocytosing a bacterial cell. It fuses that phagosome with a lysosome, and those lysosomal enzymes digest this bacterium. And then those antigenic components of the bacterium are actually placed on these receptors on the surface of the cell. And the cell runs around and shows these ligands, if you will, to other white blood cells and says, hey, this is the bad stuff. Kind of a tangential slide aside to this is this. You know, back in the COVID days, what we were doing is we were introducing an mRNA into our bodies. And that mRNA coded for a spike protein on SARS-CoV-2. So one of these spike proteins, we made an mRNA for it, put it in our bodies, it got in our cells, and it hooked up with our ribosomes. And our ribosomes translated it into a spike protein. And then what happened was we did this. Our white blood cells gobbled it up and put it on their membranes like this and showed the spike protein to other white blood cells and said, hey, why don't you make antibodies to this? And now you got antibodies that can bind to spike proteins. So it's just a really interesting, beautiful, actually, mechanism of stimulating our immune system to generate a response rapidly if you came in contact with that viral antigen again. All right, membrane potential. I'm going to give you just some basics here, but we'll go into more detail, obviously, when we get to neurophysiology. So I said cells are more negative on the inside than the outside. And that potential difference across the membrane, we consider to be a transmembrane potential. Does that make sense? So there's a TMP, this voltage across the cell. Now, it's not unlike if you took a voltmeter and hooked up a positive end and a negative end to your positive and negative electrodes, and you would hope to read 12 volts. The difference is cells are much smaller, so we use what's called a millivoltmeter. And if we took a positive electrode and we put it on the outside of the cell and a negative and put it on the inside of the cell, and really it's not like a positive and a negative per se, just one electrode on the outside, one on the inside, we'd actually read a voltage difference across cells. It's about 70 millivolts. Now, if you were to take one of these electrodes and let's say you would put it here and the other one you would put here, you wouldn't measure any difference. So the difference is just right across the plasma membrane. So here's your plasma membrane, and you got some positive charges here and some negative charges here. But if you went deep into the interior of the cell and way out here in that solution, you wouldn't measure any difference at all. So right across the bilayer. So cells more negative on the inside than the outside are said to be polarized. So they have this polarity. And they're referred to as excitable cells. Pretty much every cell of the human body is like this. The transmembrane potential when a cell is at rest, in other words, not really doing anything, would be its resting membrane potential. And those values that we need to know later for neurons will be minus 70 millivolts, and for muscle cells will be minus 85 millivolts. Now, the minus really doesn't exist. It's just a reference. In other words, what we're saying by this is 70 millivolts, but the minus just means more negative on the inside than the outside. So it's in reference to the solution. Cell signaling. So cells can communicate with themselves. They can release ligands that bind to their own receptors. We call that autocrine communication. Cells that are in the same tissue or the same organ can release chemicals and interact with each other. We call that paracrine. Para means next to, paracrine communication. And then there's cells that communicate over long distances with other cells through the bloodstream. We call that endocrine. There are cells that communicate over long distances, but not through the bloodstream, through these axons with synapses, and we call that neural communication. There are cells that communicate with each other by gap junctions that are in their membranes. So there's pores in the membrane that connect cells together. Small molecules and ions can move across. Another term for gap junction is a nexus. So that is why that term's there. Good example of this would be like a sheet of smooth muscle cells. All these smooth muscle cells are connected by gap junctions. And then direct contact would be basically what we just talked about with antigen presentation and stuff like that. Hey, here's a SARS-CoV-2 spike protein, and then here's a receptor that binds to it, something like that. So direct contact. Direct contact could also be cells binding to cells to form us, multicellular organism. So here's some examples. Here's direct contact. That's a really nice scanning electron micrograph of white blood cells and tumor cells. And really you have one tumor cells with a bunch of white blood cells that are trying to attack it. And again, their membranes are not touching. You don't have to know all this junk here, but here's the membrane of the tumor cell. Here's the membrane of the white blood cell. It's all of these proteins in the membrane that are touching each other. It's not the membranes. So that's direct contact. Here's nexus communication. So we have cardiac cells or smooth muscle cells. Here's one plasma membrane. Here's another. And then this right here is called the gap junction. So a gap junction is made up of thousands and thousands of gap junction channels. So this is a gap junction channel, another one. And there's two hemichannels that make up the channel. Here's one hemichannel in one membrane. Here's another hemichannel in the other. The hemichannel is called a connexon. And each hemichannel has 6 proteins that make it up. 1, 2, 3, 4, 5, 6. All those individual proteins are called connexins. So 6 connexins make up a connexon, make up a hemichannel, and 2 hemichannels make up a gap junction channel, which make up an entire gap junction. Thousands of them make up an entire gap junction. Paracrine, local communication. So let's talk about this nice example here. You got some bacteria that get into a wound that you have. And now what happens is you have some cells hanging out here like mast cells and macrophages. And what they do is they respond to this by releasing chemicals that we call cytokines. And these cytokines are working in a paracrine fashion. So what they're doing is they're activating other white blood cells and stuff like that. But what they're also doing, like the mast cell is releasing some cytokines that are causing your blood vessels to be leaky. One of them is called histamine. So histamine is causing the blood vessel not really to vasodilate per se, but it's actually causing the endothelial cells to kind of move away from one another and create these gaps. So if this was a blood vessel, here's one endothelial cell. Normally they're like this, but now it's causing gaps between the endothelial cells. And what that does is that causes fluid to leak out, but it also helps white blood cells crawl out. And you can see that here. So white blood cells crawling out. So look, white blood cells are in your circulation, but they don't do anything there. If they did anything in your circulation, you'd be in an ICU. You would be in what we call sepsis or septicemia. So white blood cells are just using your bloodstream to get where they have to go. which more often than not is in your skin or in the wall of your GI tract, wherever you have an infection. But they're circulating around. How do they know where to go? Well, these white blood cells are leaving a popcorn trail. So they're dumping these cytokines, and now the red blood cells say, okay, I need to slow down here and crawl out here and start phagocytosing these bacteria. So what you get is the cardinal signs of inflammation here. So for example, you get something that's called tumor. You get tumor, which is swelling. And you get swelling because all this fluid is coming out with these white blood cells. So now think about when you have an infection. The site of infection is swollen, right? So we call that tumor. Also, you get calor. It feels warm to the touch. It feels warm to the touch because plasma is coming out. And the plasma is carrying a lot of heat. So we get calor as well. We also get some dolor. We get some pain. And we get that pain because that swelling is pushing on neurons. And when it pushes on neurons, it activates these pain neurons. So we get that as well. And then we get this reddish color as well. We call it rubor. And that's literally because we have plasma right beneath the surface of the skin. Plasma is leaking out of our blood vessels. And all this is great, right? If you go to the doctor and your elbow is swollen and it's warm to the touch and it's red in color and it's a little bit painful, they probably say, go home, this look good. It's healing. But what we do is we take antihistamines and stuff like that because we don't like the pain and we want to try to reduce the inflammation and stuff like that, which is fine. But it does, think about it, it does actually counter the good things that are going on. These are all positive things that are happening to promote white blood cells to get to that site of infection to fight off that infection. Endocrine communication. Yeah, great. We're going to go through this in great detail, but you have cells in the body somewhere. Maybe they're in your pituitary gland in your brain, and they're sending hormones way down to your adrenal glands sitting on top of your kidneys. So it's very long distance communication, and generally we like to say that it's long lived. So neural communication is very short lived, a couple milliseconds. This is much longer lived, and it's more widespread. So these endocrine molecules can reach many cells of the body, whereas a neuromuscular junction, for example, only one cell can be affected. A synapse is only affecting one cell. So that's neural communication. So, or really that's the compare and contrast of endocrine and neural. Any questions before I go to this?
I have a quick question.
Yeah.
You were saying on the paracrine communication slide with the whole diagram, you were saying white blood cells, but I don't see anything labeled. Were you talking about the blue thing escaping the blood vessel?
Oh yeah. So a neutrophil is a white blood cell. Mast cell is macrophage. All those are white blood. And the white
Okay.
blood cells I was talking about cruising around in your bloodstream are right here. Yeah, the blue
Okay.
cells.
Alrighty. Thank you.
Yeah. Sorry for assuming that. Yeah, so those are all white blood cells. So we got these receptors, proteins expressed by cells. They're either in the cell membrane, so we call them plasma membrane receptors or cell surface receptors. Then we also have ones on the inside of the cell. We call them intracellular receptors. What binds to these receptors are ligands or ligands, either way you like that. These are signaling molecules or chemical messengers. They could be neurotransmitters, they could be endocrine hormones, they could be neuromodulators, they could be a lot of things. Generally speaking, for this class, they'll be neurotransmitters. You should know the difference between endogenous, exogenous. So endogenous would be something native to the human body. So it's a chemical, it's a ligand that's normally in your body, but maybe you're going to take a drug to try to block a receptor. That would be something exogenous. So you're taking something from the outside of the body and putting it into the body. So there's a cell surface receptor. You should assume that if there's cell surface receptors, that means the ligand is hydrophilic, so it can't get into the cell. And then here you can see that the ligands are actually going right through the membrane. So those are going to be hydrophobic ligands, like maybe a steroid molecule. And then they bind to receptors that are either located in the cytoplasm. So we'll call that a cytoplasmic receptor. Some of them are right in the nucleus. So we call it a nuclear receptor. And then the hormone receptor complex will basically act as a transcription factor and augment DNA function. Okay, intracellular receptors. So we have hormone receptor complex alters gene expression. Plasma membrane receptors, they can be enzyme receptors, so like a protein kinase. You could have ligand-gated ion channels. We call those ionotropic because when the ligand binds to the receptor and it becomes a channel permissive to ions, that's ionotropic. Now, most of the receptors in the human body are not ligand-gated. Most of them are G protein coupled receptors. Let me take that back. Most of them are not ionotropic. Most of them do bind ligand, but they're not ionotropic. In other words, most receptors, a ligand binds to it, and then the protein just undergoes a conformational change. Doesn't become a channel. And we call those G protein coupled receptors or metabotropic. Those are the most common in the human body. So the most ubiquitous receptor. How many people have heard of, oh, you got these enzymes that, or these transporters, sorry, these receptors that they span the membrane 7 times?
I have.
Yeah, the seven pass transmembrane receptors. So yeah, so that's the G proteins there. The G protein coupled receptors. So what it's attached to is a G protein on inside the cell, and we'll talk about that in a little bit. You should know the difference between an agonist and an antagonist. So an agonist is something that activates a receptor. So it could be the endogenous chemical, or it could be some exogenous chemical that you've created to agonize the receptor. And then an antagonist is something that blocks. So you may have heard of a drug like a beta blocker to lower blood pressure. Well, they bind to certain receptors in the body that normally bind norepinephrine and epinephrine, but when these bind, they block the channel from the binding of norepinephrine and epinephrine. And we'll talk about those in great detail. So sometimes they're called blockers instead of antagonists. You've probably learned about upregulation and downregulation. So if you have a cell here that's got hormone on the outside of it, it's got receptor, it wants to increase its responsiveness to the hormone on the outside of the cell, it can manufacture and put more receptors in the membrane. So we call that receptor upregulation. Flip side is if it wants to reduce its response to hormone, it can downregulate its receptors. And so now its response is mitigated. This is partly responsible for what we know as drug tolerance. Yeah, it's pretty rough.
So these that work in pain receptors.
Yeah. Yeah, it's not the only mechanism involved in drug tolerance, but certainly it's one of them. Signal transduction. So pretty important here. So we have a ligand binding to a receptor. The receptor changes in conformation, and that receptor was already interacting with a protein, and that causes that protein to change in conformation, and that causes the next protein to change in conformation, which causes the next to change in conformation. So you get this whole cell signaling pathway that goes on. And generally what's happening, as I mentioned before, you have kinases and phosphatases that are being activated. A kinase would be, for example, it's possible this receptor is a kinase itself, and it puts a phosphate on this protein. And then this one puts a phosphate on this protein. And then maybe this one takes a phosphate off this one. So you get these dephosphorylations and phosphorylations. That's cell physiology. That's what's causing things to happen in cells, turning things on, turning things off with phosphates. So a kinase phosphorylates something, a phosphatase removes a phosphate. So we can call this the first messenger. These are second messengers here. So here's a list of second messengers that we're going to go through. So calcium itself is a second messenger. Cyclic AMP is. Cyclic GMP is. Diacylglycerol, we'll talk about that. And inositol triphosphate. These are the major ones we'll talk about. We'll also see that very often calcium binds to cyclic AMP. All right, take that back. Calcium binds to calmodulin.
Is PKA one, or is that like a third messenger?
Yeah, it's so
I know cyclic GMP activates
so
that,
it
I
won't
think.
be it won't be a second messenger. I don't what I was thinking about is if we consider it a third messenger. Maybe some people do. I've never heard it, but yeah.
I think they're
It'll
all
be
just referred
activated
to as second messengers, aren't they? Like even if they're
hold
like the
on.
third in a chain?
I'm sorry.
I thought that they were all referred to as second messengers, like if they came after the primary, like direct signal response, like anything else was
No,
called a second
no,
messenger.
no, PKA wouldn't be a second messenger. These are the second messengers here. Whether or not PKA is considered a third, I don't know. I've never heard of that, but makes sense, but never heard of it. But calcium often binds to calmodulin, so that calcium calmodulin complex is also a second messenger that we'll see. So nothing really to memorize here, but we're going to talk about these second messengers as we move forward. Well, I guess it is time to memorize them. And we could save this for neurophysiology, or you could learn it now. Either way, when we get to neurophysiology, you're going to have to know this. So I'll just go through it now. So these are 3 G protein coupled mechanisms. Now, this is the receptor, this is the G protein. I don't care that you know norepinephrine or what the name of this receptor is. It's not important right now. What's important are the 3 different types of G proteins. There's a G sub S that stands for stimulatory. There's a G sub I that stands for inhibitory. And there's a G sub Q that nobody knows what it stands for. I mean, they really don't. Someone ran out of letters or something like that. So G sub S, G sub Q, G sub I. Now, what I want you to know is these signal cascades here for each different one of these. Now, it may look daunting right now, but really 2 of them are very similar. So let's take a look. When a ligand binds to a receptor and activates a G sub S protein, that G sub S activates an enzyme called adenylyl cyclase. Now, that protein is actually in the membrane. Maybe you've heard it as adenylate cyclase. Same thing. It's in the membrane, but they show this as a flow chart, so that's fine. But it's really it's in the membrane. Adenylate cyclase is going to convert what to cyclic AMP?
ATP.
You got it. ATP to cyclic AMP. Now, cyclic AMP is your second messenger, and cyclic AMP is going to activate protein kinase A, which we often call PKA. And PKA is going to run around and phosphorylate things. So PKA will increase phosphorylation in the cell and let's say in general, increase the activity of the cell. Now, the flip side of that is over here. And this is a G sub I. So ligand binds to receptor, receptor activates an inhibitory protein, and that inhibits adenylate cyclase. So now your cyclic AMP levels are down, your protein kinase A activation is down, and you got less phosphorylation of protein. Now, in order for cyclic AMP to go down, you can't just inhibit adenylate cyclase because it would just be at a certain level. But yeah, cyclic AMP will eventually be degraded to AMP. So it can. So really just inhibiting the enzyme is enough for cyclic AMP to drop. So if you know one pathway, you know the other here. The third one is a G sub Q. And the G protein Q activates PLC, phospholipase C, and that is in the membrane as well, just not shown here. And phospholipase C is actually going to cut one of your phospholipids up in your membrane. Now remember, that looks something like this. So you have this, I take that back. You have this polar head group and you have these two fatty acids. This will look something more like that. Whatever. So it cuts that up. And what it cuts it up into is this DAG, which is this, a diglyceride or diacylglycerol, and then this, that phosphate, IP3, so inositol triphosphate. What DAG does is activate PKC, which is akin to pKa, runs around and phosphorylates things. And then what IP3 does is it opens up calcium channels in your membrane, lets calcium come into the cell, but it also stimulates calcium to be dumped into the cytoplasm from some smooth ER. Don't worry too much about that right now, but smooth endoplasmic reticulum can be designed to store calcium, and IP3 can cause it to be released from there as well. The end goal here is to increase calcium inside the cell, and we can talk later about what the importance of that would be. So at the end of the day, increase phosphorylation and activation of calcium binding proteins.
So both the G of S and G of Q have the same, basically have the same job, but one doesn't have calcium binding, right?
Yeah, but the proteins that they're phosphorylating are probably different.
Oh, okay, not the same proteins then. Okay.
Yeah. Okay, so we've sort of built a human body. We've gone through the cells, and so now these cells have to bind to one another to form tissue. Let's talk about how they formed first. So we had this haploid sperm cell that fertilized the haploid ovum and gave you a diploid zygote. That single-celled zygote divided by mitosis into a multicellular embryo. So that was hyperplasia. So 2 cells became 4, became 8, so on and so forth. And what also started to happen was the cells started getting bigger in size. We call that hypertrophy. So the cells are dividing and they're also getting bigger in size and the embryo is growing. But right now, every single cell is a clone of one another. But eventually, after around the 100-cell stage, they start differentiating. Some cells turn on some genes, turn off others, and vice versa with other cells. And now we get this differential gene expression, and we develop 3 what we call germ layers. So we develop an ectoderm, a mesoderm, and an endoderm. There's 4 tissues that make up the human body: connective tissue, muscle, nerve, and epithelial. So 4 tissues that make up the human body, they have to come from these germ layers. So where do they come from? So ectoderm gives rise to the entire external lining of the human body, which is an epithelial tissue. So even the cornea of your eyes, so your skin, cornea of your eyes, all that comes from ectoderm. It also gives rise to your nervous system. Your entire nervous system comes from ectoderm. So those 2 right there come from ectoderm. Now, when you look at endoderm, the entire internal lining of the human body, the lining of the digestive tract, the lining of the respiratory tract, all that becomes, and by the way, the inside lining of the uterus, the vagina, the urinary bladder, all that as well is epithelial tissue. So once again, epithelial, endoderm. And then the mesoderm gives rise to everything in the middle, which is connective tissue and muscle. So all the connective tissue of the human body and muscle come from mesoderm. Now there's two caveats here. You would think that the internal lining of a blood vessel, which is called endothelium, you would think that is coming from endoderm. Actually comes from mesoderm. So endothelium comes from mesoderm. And also mesothelium comes from endoderm as well. So you'd think these linings of these body cavities that we talked about in great detail, you would think that they're internal linings, so they should come from endo. They actually come from meso. And then the internal lining of a blood vessel, for whatever reason, also comes from meso, not endo. But everything else, every other internal lining of the body is derived from endoderm. So here are the four tissue types. So this is a nice representative area of the human body, an arm. So where would we find these four tissue types? So where would you find epithelial tissue? Well, you'd find it in your skin. Where else? Anyone?
Your intestines?
No, but by looking at just the arm here.
Oh, okay. Blood vessels?
Very good. Inside lining of blood vessels, right? So we'd also find the epithelial tissue lining the inside of our blood vessels. We'd find some epithelial tissue in our synovial joints as well, but we don't really see the joints there, but that's fine. And then the muscle, I think that's clear where we would find that, but maybe more than that. Where else would we find the muscle?
Also the blood vessels?
Very good again. So remember vasodilation, vasoconstriction. Now that smooth muscle in the wall of blood vessels, this is skeletal muscle, but hey, muscle's muscle. And then nerve fibers, we have to innervate the muscle to control it. What else do we have to innervate to control it? Say
Ligaments
it again.
or blood vessel.
Say it a third time.
The blood vessels.
Right. So we have, if you want vasodilation and vasoconstriction, you need nerve control for that. So that as well. And then the connective tissue comes in all different forms. So blood is a connective tissue, cartilage is a connective tissue, bone's a connective tissue. This is loose connective tissue that you would find in your skin. So all of that is connective tissue, and we'd find that in all those, cartilage between bones. Bone makes up, or bone is made up of largely of compact bone, but there's also some spongy bone is there. And then the blood in our circulation is a connective tissue.
I feel like I should know this, but is fat a connective tissue?
Yeah, it actually is. So
Okay.
adipose is a connective tissue as well.
Okay.
So we have these connective tissues that we call connective tissue proper, like kind of like fits the definition of a connective tissue, like a tendon, a ligament. That just seems like normal connective tissue, or connective tissue in our skin. The dermis seems like normal. Then we have this weird connective tissue, like this connective tissue that has a gel-like matrix. We call it cartilage. This connective tissue that has a fluid matrix. We call it blood. This connective tissue that has this ossified matrix, we call it bone. And then, yeah, indeed, we have adipose, which is basically a bunch of these cells with hardly any extracellular matrix at all. So we consider adipose a specialized connective tissue. So this is a tissue bed here. We said that all body cells are within 20 microns of a blood vessel, which would be a capillary. So this is really what the human body boils down to. So we have a bunch of individual cells that make us up, and all of these cells need nourishment, and they need us to get rid of their waste too. So that happens at the level of a capillary bed. So that anastomosis there is a capillary bed. We have an arterial coming in that we can vasodilate, vasoconstrict. So if we want blood to preferentially go to that capillary bed, we can vasodilate it. And now what we can do is start diffusing like this. So you can just follow that flow there from the IVF, meaning from the plasma, to the ISF between the cells into our cells. And then on the venous end of this capillary bed, we can do the reverse, from the cells back to the ISF, back into the vasculature. And from the venule, you can go back to the liver and then you can detoxify all these things and remove these things from the body and basically regenerate the circulation, if you will. But not everything will go through the liver. Any questions on that? By the way, we do have lymphatic vessels located in our tissue beds as well. When you're doing all of this, what would be called filtration here and reabsorption here, there's going to be a net loss of fluid. And when that fluid is lost, it builds up in this space and it could cause swelling, which is called edema. Well, it normally doesn't because these lymphatic vessels, they pick up all that extra fluid. The lymphatic vessels actually begin here in the tissue bed. They pick up all that fluid and they take it back to your cardiovascular system. And we'll talk about where they dump it later. All right, so lastly, we have these cell-cell junctions. What holds cells together is something that's called a junctional complex, and here it is. Here's another look at it here. This is an actual image. That's a transmission electron micrograph. And then just an illustration to the left of it. So here we have most apically a tight junction. We also call that an occluding junction. We have an adhering junction that we also call a belt junction. And then we have a desmosome that we also call desmosome. So really, the ones that hold cells together are these two. Tight junctions don't hold cells together. Kind of weird, right? Sounds like it would, tight junction. But really what it is integral membrane proteins in this membrane, integral membranes proteins in this membrane. And what they're doing is they're basically obliterating that space there. That intracellular space between the cells, they're basically blocking anything from moving that pathway. If you want something to get in your body, you want it to go through your membrane. You don't want it to go between your cells because you can regulate with channels and transporters and all that stuff what gets in. You don't want things to get between your cells. So we block that space. We occlude it with occluding junctions. These guys hold cells together. So the belt junction has integral membrane proteins touching integral membrane proteins, and they're connected to actin filaments of your cytoskeleton. And then the desmosomes have integral membrane proteins connected to integral membrane proteins connected to intermediate filaments that course throughout your cells. They basically dissipate forces that would be that would lead to cells being torn apart. For example, if you pulled on that microvillus this way and pulled on this microvillus this way, this kind of looks like a zipper. It would look like, well, you could unzip this and unzip that. The reason cells don't unzip is because as soon as force is applied to these receptors, it's transduced through these filaments. And so it reduces those shearing and tearing forces. So that's the junctional complex. Also on the lateral side of cells, we do find gap junctions, but please note that they're not part of a junctional complex. They're communicating junctions. They allow cells to communicate with one another. ATP, ions, stuff like that can cruise through these junctions.
Do cells
And
signal?
then, one last thing, sorry. And then you need to bind the cells to the connective tissue of the body. So then we take half of a desmosome to do that. So we call that a hemidesmosome. And now that just lags down to connective tissue fibers. And now we're lagged down to the body. Yeah, go ahead.
I was asking, do cell signaling happen through these junctions as well?
Say that again.
Does cell signaling happen through the junctions?
Yeah, that's a great question. It is really a great question. I don't know, is the short answer. I wouldn't be surprised to learn that it does. So the question was, does cell signaling happen through these junctions? I know nothing about it, to be quite honest with you. I think that the question is stupendous. I love the question. I would imagine that somebody's researching this, and it may have been a while ago, someone's been researching this, And people have realized that stress on cells can cause activation of proteins in cells and stuff like that, or deactivation. So in that sense, I would have to believe the answer is yes.
All
So
right.
for example, you know, if you have a piece of bone, so let's say here's a tubercle on a bone, here's the head of the bone, right? Say it's the humerus. That tubercle right there is connected via a tendon to muscle, right? Well, if you repeatedly stimulate that muscle to contract, that's going to pull on that tendon. And actually, that'll cause remodeling of the tissue there. Osteoclasts and osteoblasts signals will be all over the place, causing these cells to remodel the bone to build up that tubercle in the direction of that stress. So I would have to believe that signal